A very good morning to all of you. So today's our topic of discussion is that of atomic absorption spectrometry. Yesterday we have already studied what is the use of this atomic absorption spectrometry, what is the principle and what are the applications. In today's session we will study the instrumentation of this atomic absorption spectrometry. Okay. So what is the instrumentation? First thing is there is a hollow cathode lamp. So there is a hollow cathode lamp, and inside this hollow cathode lamp this is the source of your light source. From here light is generated and it contains metal with inert gases like your helium. Helium is filled over here, okay, because helium is inert gas, so it cannot interfere, okay. And what is first is that of hollow cathode. This is your hollow cathode lamp and it is a light source. Being a light source, what is the function of this hollow cathode lamp? It produces radiation of desired frequency. Okay, so main function of this hollow cathode lamp is it is a light source provider. Okay, next comes your chopper. Second is your chopper. This is your chopper. So what is the function of this chopper? This function of this chopper is it will modulate the intensity of light. From this hollow cathode lamp, light passes and first this light enters into your chopper. So here when it enters into your chopper, then this chopper will modulate the intensity of light. Okay. Then third, include your this sample cell. This whole thing is known as sample cell, and this sample cell contain a part that contains your sample. This sample in which you are willing to detect any your desired metal. Okay, and this sample in which form? This sample in your liquid form. And this also contain a atomizer. This is what? This thing. This is your atomizer. And in this atomizer, there is on upside of atomizer, there is a burner in which flame is burning. Okay. So this whole setup is your sample. Yeah, you can say sample chamber also you can say. Okay. Now what is the individual function? In this sample chamber, there is a sample cell which contains sample. And sample is in which form? Sample is in your liquid form. Okay, this sample which contain your desired metal for which you are going to do your atomic absorption spectrometry. And what is the function of this atomizer? The function of this atomizer is that this atomizer converts your liquid. This atomizer, the sample which is in liquid form, this atomizer convert your liquid sample into your gaseous form. As we studied yesterday, what is the principle of this? Thus liquid is first we are just heating liquid in the flame, and in that liquid it contains solute as well as solvent. So when you will heat that solution, then solvent will evaporate and there is remaining of your what? Tiny particles of your solute. So the function of this atomizer, it will convert your tiny particles into your gaseous form, and then that gaseous comes into your atom form, and then that atom is in your unexcited or ground state, and that will absorb the radiation, and from when atoms which are in the ground state, after absorbing the radiation, goes into excited state. Okay, yesterday already we have studied in our principle, and that amount of radiation which are taken by that particular unexcited atom is measuring your concentration. So amount of radiation is directly proportional to your concentration. Okay. Then what happened after it? This flame is there, which is act as a heating purpose. Okay, and after that what happened? What is next? So this is your hollow cathode lamp is your first. Then second is your chopper, and third is your this whole sample chamber which contain your sample cell and your atomizer, and your burner is here where this flame is burning. And next fourth is your that of lens. What is the function of this lens? This lens is simply passes the light. Okay, your light is passes and it will simply help in the passing the light. Next, fifth one is your monochromator. This is your what? This is your monochromator. What is the function of this monochromator? This monochromator receives the light, polychromatic light, and being a monochromator, it will convert your polychromatic light into your monochromatic light, and you will get your desired frequency wavelength. Okay, so this is the function of monochromator. In whole biochemistry of instrumentation, wherever you will read the monochromator, in everywhere the function of this monochromator is to convert your polychromatic light into monochromatic light and of desired frequency in every instrumentation. Okay, so remember this always, the function of monochromator is to convert your polychromatic light into your monochromatic light, and from there you will get your desired frequency wavelength of light. All right. After that, 6th one is your detector. From here it will pass into the detector. So detector will what do? The detector will detect your signal. The signal which are coming from this monochromator, this detector will detect, and the signal which are coming from this monochromator in the form of heat energy. And the function of this detector is also that it will convert your signals of heat energy in the form of electrical energy. Okay, it will convert heat energy signals in the form of your electric energy signals. Got it? Because you cannot measure the heat energy signals, you can only measure electric energy signals. So detector will detect the signal and it will convert your heat energy signals into your electric energy signals. After that, it will throw out to the amplifier. What is the function of amplifier? As name suggests, amplifier, so it will amplify the signals. The signal which it received from the detector, it will amplify that signal so that we can get better reading and better result. After that, it will pass from your readout device. Readout device is that on which instrument you will get your reading. Anywhere the readout device is that in which you will get your final reading. Okay, so this readout device give you reading and you will just take that reading. By that reading will give you quantitative estimation. Alright, so this is whole about your instrumentation of atomic absorption spectrometry. It is an important topic of the paper 4, and question is asked as that what is atomic absorption spectrometry? What is the principle of atomic absorption spectrometry? Write its application and instrumentation. So in this question you will read, you have to write all these things. First you will write the use, then you will write principle, after that your application, and you just make this diagram and you can write it simply in this form also. So you will get the good marks. Okay, and it is frequently asked in a short note. No, never ever long question come on this topic. So it is asked as a short note, as either as a 5 marks question or as a 10 marks question. Okay, so that's all for today. Next session we will read another instrument.